you. Today we are going to take our next topic, biofertilizer and their role in crop production. See, what is meant by the biofertilizer? The preparation containing live or latent cells of efficient strains of nitrogen fixing, phosphate solubilizing, and cellulitic microorganisms used for the application to the seed, soil, or composting areas with an objective of increasing number of such microorganisms and to accelerate the certain microbial processes to augment the extent of availability of nutrient in a form which can be easily assimilated by the plant. See, biofertilizer is the preparation which contains the different types of the strains. For example, nitrogen fixing bacteria, phosphorus solubilizing bacteria, then sulfur cellulitic microorganisms which are used for the application to the seed at the time of the sowing, thereby increases the number of microorganisms in the soil and increases the availability of nutrient in a available form. This is the definition of biofertilizer. Next is the classification of biofertilizer. There are three classes of the biofertilizer. First is nitrogen fixing microorganisms. Second is phosphate solubilizing microorganisms. Third is sulfur solubilizing microorganisms. Nitrogen fixing microorganisms, symbiotic nitrogen fixation and non-symbiotic nitrogen fixation. Which are the symbiotic nitrogen fixing microorganisms? Rhizobium, Azolla, Frankia, Benzyrhynchia. All these are the examples of the symbiotic nitrogen fixing microorganisms. Non-symbiotic microorganisms are Azotobacter, BGA. See, the various types of biofertilizers which helps the plant to grow at the different level of its growth rate are nitrogen fixing biofertilizer, phosphate, under that phosphorus solubilizing biofertilizer, and phosphorus mobilizing biofertilizer. Then biofertilizers for the micronutrients, then plant growth regulator or promoting rhizobacteria, then compost. In that, nitrogen fixing biofertilizer increases the soil nitrogen level. Then it fixes atmospheric nitrogen in the soil and make it available to the plant. This is the main role of the nitrogen fixing biofertilizer. Examples are Azotobacter, Nostoc, Rhizobium, Azospirillum. Next is the Azotobacter. It is a free-living nitrogen fixing bacterium are found in the non-leguminous plant. Next is cyanobacteria, that is Nostoc, blue-green algae. It is a free-living as well as symbiotic. They also fixes the atmospheric nitrogen to the soil. Rhizobium is the symbiotic nitrogen fixing bacterium found in the leguminous plant. Azospirillum, it is an associative symbiotic nitrogen fixing bacterium and found in the gramineous plant. Next is the phosphate biofertilizers. Phosphorus solubilizing biofertilizers and phosphorus mobilizing biofertilizers. In that, first one is the phosphorus solubilizing biofertilizer. They solubilize the insoluble phosphate from the organic and inorganic phosphate sources. Then it releases the insoluble phosphorus in soil and fixes in the clay mineral. Then it secretes the organic acids and lowers the pH to dissolve the bound phosphates present in the soil. The example is the species of Bacillus, Pseudomonas, Penicillium, and Aspergillus. Next is the phosphorus mobilizing biofertilizer. They transfer the phosphorus from soil to the root cortex. For example, arbuscular mycorrhiza, AM fungi. This fungus penetrates the cortical cells of the roots, thereby increases the surface area of the root, then displays the absorption equilibrium of phosphate ions, which increases the transfer of P ions. Then it stimulates the various metabolic processes. Then arbuscules absorb these nutrients into the root system. Biofertilizers for the micronutrient. The bacterial species are silicates and the zinc solubilizers. Degrades the silicates and the aluminum silicates in the soil and helps in silicates weathering. Example, Bacillus species. Plant growth promoting rhizobacteria, PGPRS. It acts as both biofertilizer as well as biopesticides. Promote growth by improving the nutrient availability, then suppression of the plant diseases and the phytohormones production. We'll see one by one different microorganisms. Rhizobium, it belongs to the family Rhizobiaceae. It is a symbiotic in nature, associated with the legumes only, then enters into the plant root through the root hairs. Pink color of nodule due to the leaf hemoglobin present on the root where rhizobium is present. Example is the soybean Rhizobium japonicum. It fixes up to the 50 to 100 kg nitrogen per hectare with leguminous only. It is useful for the pulse legumes like chickpea, red gram, pea, lentil, black gram, oilseed legumes like soybean, groundnut, forage legumes like berseem, lucerne. Okay, the seed treatment of rhizobium is given at the rate of 25 gram per kg of the seed. Azotobacter, it belongs to the family Azotobacteriaceae. Heterotrophic in nature, it is a free-living, non-symbiotic in nature. It fixes 20 to 40 kg nitrogen per hectare, then produces growth-promoting substances like vitamin of B group, IAA, GA3. It is used for the cereals, millets, cotton, sugarcane, vegetables at 25 gram per kg of seed. It is a non-symbiotic 20 to 40 kg nitrogen per hectare. It is recommended for the rice, wheat, millets, other cereals, cotton, vegetables, sunflower, mustards, and the flowers. Next is the azospirillum. It is associative in nature by producing growth regulating substances. Then it fixes 20 to 40 kg nitrogen per hectare. Application of this biofertilizer results in increased mineral and water uptake, then root development, then vegetative growth, and 15 to 30% increase in the crop yield. It is also recommended for the rice, millets, maize, wheat, sorghum, and the sugarcane. Associative in nature belongs to the family Spalaceae, then chemoheterotropic in nature, produces growth promoting substances, fixes up to 20 to 40 kg nitrogen per hectare. It saves the nitrogenous fertilizer by 25%, 15 to 30% increase in crop yield, rice, maize, wheat, cotton, sunflower, all these are crops. Then PSM are the microorganisms and mainly bacteria and the fungus. Saves up to 30 kg P2O5 per hectare. Also produces growth promoting hormones like IAA, GA, and the cytokinins. Bacteria are the Bacillus and the Pseudomonas. Fungi are the Aspergillus and the Penicillium. BGA, it is also the phototrophic in nature. It uses the sunlight for energy and water for the photosynthesis. They also produces growth promoting hormones like auxins, IAA, GA3. They fixes 20 to 30 kg nitrogen per hectare and they increases the paddy yield up to 20%. It is applied at the rate of 10 kg per hectare in rice field after 1 week of the transplantation. Next is the azolla. Is a symbiotic in nature, suitable only for the flooded rice, fixes up to 40 to 80 kg nitrogen per hectare, increases yield up to the 40 to 80 kg per hectare, used as a green manure, and most important species found in India is pinnata. It is recommended as green manure because of the large biomass and high iron content for the submerged rice field within maximum temperature of 38 degrees Celsius. It increases yield 15 to 20%, suitable for the flooded rice, which is symbiotic in nature. Nitrogen fixation in soil. The biological reduction of molecular nitrogen to ammonia is carried out by the certain bacteria. Are you getting? The biological reduction of molecular nitrogen to the ammonia is carried out bacteria is known as the biological nitrogen fixation. Which minerals are involved in this monophorylite, elite, vermiculite fixes NH4N on the soil? The biological nitrogen fixation is of two types, symbiotic nitrogen fixation and non-symbiotic nitrogen fixation. The symbiotic nitrogen fixation, the bacteria which lives is symbiotically with host plant, for example, legumes, soybean, groundnut, and fixes nitrogen in the root nodule. It belongs to the genera Rhizobium. Nitrogen fixed by the legume bacteria used by the legume plant or may be passed into the soil. They are live symbiotically. That's why it is known as the symbiotic nitrogen fixation. Non-symbiotic nitrogen fixation. The atmospheric nitrogen can be fixed in the soil or near to the root zone by the certain free-living bacteria and the fungi. Non-symbiotic C. Azotobacter, Clostridium species play an important role in this process. Are you getting symbiotic nitrogen fixation and non-symbiotic nitrogen fixation? Next is the phosphorus solubilizing biofertilizer. These microorganisms are mainly bacteria. Example, Bacillus and the Pseudomonas, and fungi. Example, Aspergillus and Penicillium. These microorganisms possesses the ability to bring the insoluble P into soluble form by secreting the several organic acids. These microorganisms have ability to bring the insoluble P into the form of soluble one. Under favorable conditions, they can solubilize up to 20 to 30% of insoluble P and increase the crop yield by 10 to 20%. PSB is recommended for the all crop. Example is FAM or mycorrhiza. See, this type of microorganism enhances the uptake of P, Zn, S and water, leading to the uniform crop growth and increased crop yield. Also, enhanced resistance to the root diseases and improve hardness of the transplant stock. Recommended for the forest trees, forest grasses, maize, millets, sorghum, barley, and the leguminous crop. Sulfur oxidizing microorganism. The bulk of soil sulfur is in organic form, which is metabolized by the soil microorganism to make it available in an inorganic form for the plant nutrition. In the absence of oxygen, certain microorganism produces H2S from organic sulfur substrates. Example is bacteria Thiobacillus species, actinomycetes, fungi Aspergillus penicillium. Thiobacillus produces H2SO4 when elemental sulfur is added to the soil. Rate of application of biofertilizer is seed treatment of biofertilizer at 25 gram per kg of seed and cellulitic fungus at the rate 1 kg per ton of the crop residues. Standards and the quality control. The quality of biofertilizer means the presence of the right type of microorganisms in active form and the desired number. It is known as the quality of the biofertilizer. It has to be controlled at the various stages of production, marketing, and the storage. During mother culture stage, then during carrier selection, during broth culture stage, while mixing of broth with carrier, during packaging, and during storage. Care should be taken while the preparation of mother culture and broth culture that there is a no contamination. Right type of carrier should be select having the high water holding capacity and it must be passed through the standard sieve. pH of the culture should be maintained between 6 to 7.5. Expiry date of Rhizobium, Azotobacter, PSB, Azospirillum is 6 months from the date of manufacture. All the biofertilizers must be stored in the cold space. The desired number or count of microorganisms must be maintained throughout the 6 months. 10⁻⁷ per gram of carrier of Rhizobium and 10⁶ per gram carrier for the Azotobacter. The microbial strain or particular species of microorganism should be checked time to time. Methods of application of biofertilizer. There are different methods: seed treatments, then seedling inoculation, set inoculation, in that pasting of birds, then sugarcane set inoculation, soil inoculation, and the seed pelleting. What is the contribution of biofertilizer in agriculture? They supplement the fertilizers for meeting the nutrient seeds of the crop. Then they can add 20 to 200 kg nitrogen per hectare under optimum condition and stabilizes the or mobilizes 30 to 50 kg P2O5 per hectare. Then they can liberate the growth promoting substances, vitamins, and helps to maintain the soil fertility. Then they suppresses the incidence of the pathogen and control diseases. They increases the crop yield by 10 to 50%. Then they are cheaper and the pollution free. They improve the soil physical properties, tilth, and soil health in general. This is about the biofertilizer. Next is about the fertilizer control order. An order issued by the Government of India under the powers conferred by the Section 3 of Essential Commodities Act 1955. Under the powers, the government controls the production, marketing, price, and the quality of the fertilizers. There are total 39 clauses of this order covering the definitions, prices, registration of dealers, then fertilizer mixture, regulation on manufacture, sale, packaging requirement, disposal of non-standard fertilizers, then enforcement authority, analysis of sample, etc. The schedule first of the order includes detailed specification of fertilizers covered by it. The schedule first of the order includes detailed specification of fertilizers covered by it. In schedule second, detailed procedure regarding the sampling technique and method of analysis is given. Here, about the urea, moisture percent by weight and the maximum specification is 1. The total nitrogen percent by weight on dry weight basis is 46. Biuret percent by the weight 1.5. Then particle size not less than 90% of material shall pass to the 2.8 mm sieve and not less than 80 shall retain on the 1 mm sieve. This is about the particulars and the specification of ammonium sulfate. This is the particulars and the specification of the SSP. This is the particulation of the MOP. This is the assignment. Solve all these assignment in your assignment notebook. Write down the role of biofertilizers in agriculture. Then define biofertilizer, give its type with suitable example. Write down the nitrogen fixation, explain its types. Give the contribution of biofertilizers in agriculture. Then write down the standard and the quality control of biofertilizers and explain fertilizer control order. Okay, today we will stop here.